We have completed until 5 zone model in previous class. Let us start part 2. Types of root nodules. There are two main types of root nodules. They are determinate nodules, indeterminate nodules. These determinate root nodules have limitations. They grow until to a certain limit and they stop growing. After growing to a certain limit, the meristem zone stops growing. In this type of root nodules, cell division will be stopped after certain limit. So only cell elongation is happens. Okay. So cells grows from small to large. There is no division. So growth happen by enlarging the cells. Now see the characteristics. They are in spherical shape. Growth is limited. Okay. Now let's come to indeterminate nodules. They are in cylindrical shape. Here growth is continuous. So there is no stop in the growth. Also cell division happens here. It happens in P, that is Pisum sativum. Okay. The another one is clover. Determinate nodules happens in soybean as well as in groundnut. Let's go to next slide. Regulation of nitrogen fixation, autoregulation of nodulation, that is AON. So what did you understand from the heading? If there is excessive nodules are formed, they have to be stopped the further growth. Okay. So it happens by autoregulation of nodulation. Why it should be happen? Because it consumes excessive energy. If there is sufficient nodules, why the plant needs to produce further nodules? It can save its energy, right? Let us see how the mechanism works. Autoregulation of nodulation, that is AON. It works like hormones. What happens in hormones? Let us see in humans. Brain produces hormones. They are travel through blood and reaches to the targeted specific cells. So it is happen in plants also. Okay. It is a long distance signaling mechanism. It happens between roots and shoots. Okay. It limits the nodule formation. So how it works? If there is a root infection, signal sent to the shoots, that means stems. These shoots sends back inhibitory signal back. So nodulation is reduced. So what did you understand here? Due to the root infection, nodules are producing excessive. So signal is going to shoot taking the action. Okay. If the nitrate is excessive in the soil already having the nitrogen. So why plant need to produce further nitrogen? So it won't produces. The second thing that controls nitrogen fixation is oxygen concentration. If there is excessive oxygen, so nitrogenase enzyme won't work. So now nitrogen fixation will be stopped. Plants evolved to maintain oxygen levels by leg hemoglobin protein. So it maintains low oxygen levels so that rhizobium bacteriums get the oxygen and also it prevents the oxygen to the nitrogenase enzyme. Nitrogen fixation needs energy that comes from the photosynthesis. So there is good light in the day and photosynthesis is high, then nitrogen fixation also high. So it depends on photosynthesis. Regulation of nitrate reductase. What is the role of nitrate reductase? Did you remember about it? It converts nitrate to nitrite. I told you earlier. Okay. Why it should be regulated? I told you earlier, that is nitrite is dangerous to the cells. If there is excessive light, this nitrogen reductase produces more. So its activity is high. If there is darkness, this nitrogen reductase activity is low. Okay. Nitrogen activity will be more if there is excessive sugar. Okay. So sugar and light both increases nitrogen reductase activity. What is nitrate reductase? I told you it converts nitrate to nitrite. Okay. By using NADH, so it formed nitrate as well as NAD and water. Okay. It happens in cytosol. The electron donor is NADH or NADPH. Cofactors required are FAD. Also heme, that is cytochrome B557. Molybdenum cofactor, because this is the rate limiting step. So plants regulate the nitrate reductase in multiple levels. These are the levels: transcriptional regulation, post-translational regulation, enzyme degradation. If there is excessive light or if there is excessive glucose, okay, this nitrogen reductase will be more. I told you earlier. Okay. Nitrate itself acts as a signaling molecule. So it motivates the production of nitrate reductase if there is excessive light, if there is excessive glucose. It also lowers the production of nitrate reductase if there is darkness or if there is less glucose. Okay. How it happens? First NR, that means nitrate reductase genes activated. Then mRNA synthesis rises. Then nitrate reductase produced more. If there is low light, low glucose, it will not be produced. Okay. The second regulation happens with post-translational regulation. It happens rapidly. That means it happens in minutes. Okay. Nitrate reductase has serine residue. Phosphate group added to this serine residue, so it stops the production of nitrate reductase. If this phosphate group is removed, then nitrate reductase will be activated. The third regulation is happened by enzymes. Nitrogen reductase have short life, so it will be degraded after its life, right? And also new nitrate reductase synthesis will be stopped. Environmental factors that affects the nitrogen reductase production. Okay. The first one is nitrate availability. If there is excessive nitrate, then nitrate reductase will be more. Why? If there is no nitrate, plant need to produce nitrate. It requires more energy. So plant have to waste its energy. So in order to do that, if there is excessive nitrogen, then plant produces excessive nitrate reductase. The second one is light intensity. If there is excessive light, photosynthesis will be more. Then plant has more energy. Then also nitrate reductase activity will be more. This is also same point. If there is more photosynthesis, more carbon and more NR. Okay. Oxygen availability. If there is low oxygen, then nitrate assimilation will be low. Why it is decreasing in low oxygen? Because plants' mitochondria need to produce excessive energy in order to dissolve. If there is low oxygen, then there will be energy low and the production of NR will be low. The fifth point is temperature. If the temperature reaches to optimal levels, then the NR production will be high. If temperature is more, then the production will be less. If temperature is less, then NR will be produced less. We can see the summary as well. If there is excessive nitrate, then production will be high. If there is excessive light, NR activity will be high. If there is more carbohydrates, then NR expression is high. If there is more ammonium, then low NR. If there is darkness, then low NR. If phosphate bind to serine residue, that means 14-3-3 proteins. Okay. If phosphate binds here, then the production will be less. Did you remember about C2 cycle? That is oxidative phosphorylation. It happens in 3 components. The energy produced in this cycle is utilized itself. So there is no excessive production in this cycle. I told you earlier about this cycle. If you didn't watch it, please go back and watch it also. So it happens in 3 components. They are chloroplast, peroxisome, mitochondria. So why I am telling about this cycle in this nitrogen fixation? Do you know? Do you know what is the link between C2 cycle and nitrogen fixation? You can see here 2 molecules of glycogen are combining and forming serine. So what happening here? One carbon dioxide molecule is wasting here. Also one NH3 molecule is releasing out. Now you must understand why ammonia is linking with nitrogen fixation. Can you see where the ammonia is released? It is released in the mitochondria. So here photorespiration produces ammonia that is happening in the mitochondria. Where this produced ammonia goes, what is the function of it? This produced ammonia again goes to chloroplast and it enters to the GS-GOGAT pathway. I told you about GS-GOGAT pathway. If you didn't watch it until now, please go back to previous class and watch it. Okay. So this ammonia reacts with glutamate and forms glutamine. If you watch the previous class, you can understand everything. This glutamine reacts with alpha ketoglutarate and produces 2 glutamate molecules. So what is the function of glutamate? It is an amino acid. It also reacts with other amino acids and produces remaining amino acids. What are the families of amino acids? These are precursor molecules. These are amino acids. So alpha ketoglutarate produces glutamate. We have seen it. Glutamine, proline, arginine. Like that, oxaloacetate produces aspartate, methionine, lysine, threonine. Pyruvate produces alanine, valine, leucine. 3-phosphoglycerate produces serine, glycine, cysteine. PEP, phosphoenol pyruvate plus erythrose 4-phosphate produces phenylalanine, tyrosine, tryptophan. Please memorize this all, okay? This is also same diagram. Please watch it also later. So what are the functions of these precursor molecules? Let us see them. Carbon skeleton. The first one is alpha ketoglutarate. It is used in Krebs cycle. Okay. That means tricarboxylic acid cycle. Oxaloacetate. Same Krebs cycle. Pyruvate. It is the end product of glycolysis. So where these reactions are happening? They are happening in photosynthesis, also in respiration, also in Krebs cycle. Okay. The last slide that describes about nitrogen metabolism overview. Let us see what we have explained until now. Nitrate or ammonia uptake. Second step happens as this nitrate to nitrite. So we call it as nitrate assimilation. The third step is nitrite to ammonia. The second step happens in cytoplasm. The third step happens in chloroplast. Cytoplasm and chloroplast. So we call it as ammonium assimilation. Next, this ammonium reacts with glutamate, which is present earlier. Okay. So it reacts with glutamate and forms glutamine. So glutamate reacts with ammonium and forms glutamine. We call it as GS-GOGAT pathway. Also we call it as ammonium assimilation. Okay. Please remember this. This glutamate is the central molecule for producing amino acids. So it plays major role. Okay. So it forms many amino acids. Amino acids are precursor for proteins. So finally proteins are produced. Okay. I think you have understood very well by this overview. Please subscribe my channel if you like my videos. Thank you.